Good morning, dear students. Today in this video we'll study about dihybrid cross or dihybrid inheritance. Children, Mendel conducted experiments to study the segregation and transmission of two pairs of contrasting characters at a time. This cross was called a dihybrid cross. He took characters, shape and color of seeds of pea plant. So he considered round and yellow and wrinkled and green seeds. He crossed them together and got only round and yellow seeds in F1 generation, but in F2 generation, four types of combinations were observed. Two of these combinations were similar to the parental combinations, while other two were new combinations. Dihybrid cross is called so because in this we consider two different characteristics of same plant. For example, we take seeds of pea plant and consider its shape and color. In this way we get two types of seeds, round and yellow and wrinkled and green. These are phenotypes, means physical characters. Genotype or genetic composition of this phenotype is represented as round by capital R and yellow by capital Y. In other plant, wrinkled by small r and green by small y. We know that dominant gene is represented by capital letters and recessive gene is represented by small letters. Here dominant character is round shape and yellow color. Here we have taken two plants which produce seeds making genes capital R, capital Y and small r, small y and we cross them together. So parental generation is round and yellow, that is capital R, capital Y, crossed with wrinkled and green, small r, small y. Gametes formed will be capital R, capital Y from first and small r, small y from second. And now to understand more genes and traits, we can make one Punnett square and we'll understand with the Punnett square the F1 generation. So on the left hand side write the gametes of first parent capital R, capital Y, and on the top the second parent that gametes small r, small y. So in first box we get capital R, small r, capital Y, small y. In second capital R, capital Y, small y. Same way in third and fourth also. Same we get capital R, capital Y, small y. So the phenotype, this is F1 generation, genotype of F1 generation, and phenotype of this F1 generation will be round and yellow, because if capital letter is there then capital R is there means round, and any capital Y is there yellow, and all are same. So in all physical characters will be round and yellow. Phenotype is round and yellow because only dominant characters are expressed. Recessive characters are the characters which are never expressed in F1 generation. Now we take two plants of F1 generation and get them crossed together. So capital R, capital Y, small y crossed with capital R, capital Y, small y. Here gametes formed will have one gene of color and one gene of shape. So gametes formed will be capital R, capital Y, capital R, small r, capital Y, small y. And same way in another one, capital R, capital Y, capital R, small r, capital Y, and small r, small y. Now these gametes will be fused together and will make F2 generation. So we make Punnett square. We write on the left side the gametes of first parent and on the top the gametes of second parent, and then get them fused. So we get in first box capital R, capital R, capital Y, capital Y. In second one capital R, capital R, capital Y, small y. In third capital R, small r, capital Y, capital Y. In fourth capital R, small r, capital Y, small y. In this way you find out for all boxes yourself. Still I am telling what will be next. Capital R, capital R, capital Y, small y. Capital R, small y, capital R, small r, capital Y, small y, capital R, small r, small y. Same way, capital R, small r, capital Y, capital Y. In this way, you can complete this Punnett square. So, you will find that we get different types of seeds, okay, and we find that if any of the capital letter is there, R or Y, if capital R is there, so round seeds will be produced, whether it is one capital, okay, and one capital Y is there, still yellow seeds will be formed. So phenotype for these all genotype, if we see, we have round and yellow seeds 9, round and green seeds 3, wrinkled and yellow seeds 3, and wrinkled and green seeds only 1. Okay. So when you will count, you just complete this, and after that you count, so you will find these all phenotypes. So what will be phenotypic ratio? It will be 9:3:3:1. And genotypic ratio, what will be genotypic ratio? If you want to find out genotypic ratio for this, so you have to just multiply 1:2:1 to 1:2:1. This you remember. And how will you multiply? First of all, 1:2:1. Second one, 1:2:1. You multiply by each number of first ratio, like 1:2:1 पूरा जो है you will multiply by 1, then by 2, then by 1. So what you will get, do it, you will get 1:2:1 when you multiplied it by 1. Then now multiply 2, so it will be 2 1 ja 2, 2 2 ja 4, 4 2 1 ja 2. So 2:4:2. Now last one say multiply, so it will be 1:2:1. So what is the genotypic ratio for that dihybrid cross? It will be 1:2:1:4:2:1:1. That's all for today. Thank you, children. I hope you understood. Bye.